In this video, we're going to learn about one of the kingdoms from the Eukarya domain. Specifically, we'll learn about the Protists. This section covers chapter 2.3 in the Nelson textbook, so please make sure you read that section ahead of watching this video and use the guided reading questions in order to focus on the key terminology within the chapter. So as I mentioned, and we learned about previously, there are three domains under which we classify life, and then within those domains there's kingdoms. We've already learned about the bacteria and archaea domain and their respective kingdoms, eubacteria and archaebacteria. And now we're going to explore the Eukarya domain, and in this first presentation we're going to focus on one particular kingdom within the Eukarya domain, which is Protist or Protista. And I do need you to focus on those characteristics that I mentioned before for ways that we classify them, and in this presentation we'll go in more detail on these characteristics. The other kingdoms that we'll be focusing on later on are the fungi, plant, and animal kingdoms, also within Eukarya as well. It's important to really understand the different characteristics, how each kingdom is different from each other within a domain, so that you could tell which organisms are part of which kingdom and domain, and so on and so forth. So again, all living things can be classified into three domains, and each of those domains further divided into kingdoms, and those can have other divisions, but we're going to focus on the kingdom Protista. So the first thing to understand is that the most diverse kingdom in the Eukarya domain is the kingdom of the Protist. They are basically considered the everything drawer of the Eukarya domain. Any eukaryotic cell that's not a plant or an animal or a fungus will be placed under the Protist kingdom. Now, as we gather more DNA evidence and other evidence through research, eventually the Protist kingdom will be divided and broken down into maybe other kingdoms as we see which organisms are more closely related to others. But the main idea is that the Protists are the most diverse, and they're the everything drawer. So if you take a look at our tree of life here we have the ancestral eukaryote. We're strictly looking at the Eukarya domain right now. We don't have the prokaryote domain, the archaeobacteria domains here. So if you take a look, we can see the fungus, animal, and plant kingdom, and then everything else in yellow over here is considered our Protist kingdom. Again, the everything drawer. So the main point I want you to get is, unlike other kingdoms, there is no typical Protist. Because they're the everything drawer, they vary quite a bit within that kingdom. So the main thing that all Protists share in common is that they are all eukaryotes, and at the moment they're not animals, plants, and fungi. So again, they're the most diverse group, but there's no typical Protist. The main things they share in common, they're eukaryotes, and they're just not plants, animals, and fungi. Here are just some different examples of some Protists that you might see. You are responsible for being able to identify many of the organisms that we see throughout this PowerPoint and in your readings. So for example, you should know what a paramecium is or euglena is. You should also know what diatoms look like, and also we'll see some other examples as we go throughout the PowerPoint. For starters, these are great ones to know how to label. We'll go over some of the parts later on, but you should know how to label a paramecium. You should know how to label a euglena, and also what some of the different parts are for. So for example, you should know that the flagellum is used for motion. You should know that the cilia can also be used for motion. They're just more numerous and shorter than the flagellum. Flagellum is singular for flagella. So if a cell has several flagellum, we say it has several flagella. So again, a very diverse kingdom, and what we're going to do is we're going to go explore how these Protists are classified and organized. This is our ancestral eukaryote. These orange colored individuals here, or tips here, are the other kingdoms in the Eukarya domain, but all these green ones here, they are the Protist kingdom. So there's quite a few. You should know what they are. As per the presentation on bacteria and archaeobacteria, please understand that some protists are bad or harmful and some protists are beneficial. So you should know examples of harmful protist and examples of beneficial protist. So there are some that cause disease, like the Plasmodium. It causes malaria. Some protists can cause sleeping sickness and dysentery. So you should know some diseases and some protists that cause these diseases as well. So I've included a few images here, and there's some in your readings. Please make sure you go over those. And one very important point to really take away from this presentation is that most protists are actually unicellular, which actually means that most eukaryotes are actually protists, thus meaning that most eukaryotes are unicellular. So let me rephrase that. Most protists are unicellular. Since most eukaryotes are protists, what that means is that most eukaryotes are unicellular. Many people believe that most eukaryotes are going to be multicellular, but it's not necessarily the case. They're actually mostly unicellular because the most diverse kingdom, which is a protist, consists of unicellular organisms. There are some multicellular protists. We'll take a look at some examples, and there are also some colonial protists. The colonial ones are unicellular, but they sometimes like to hang out in groups. So again, below you can see the diversity that exists within the protist group. They're just so different from each other. Again, there's nothing typical about these here. It's very hard to spot their similarities other than the fact that they're eukaryotic and they're not plant, animal, and fungus. So just a few general features to go over again. So remember that protists are considered eukaryotes. And the reason they're considered eukaryotes is because they have that nucleus, the nuclear membrane that surrounds their DNA. They also have membrane-bound organelles. So for example, nucleus is a membrane-bound organelle, but they'll have mitochondria. Some protists will have chloroplast. The other important thing to understand is that protists, because they're so different, you'll find them in a variety of different habitats. Some could be at the bottom of oceans, some could be in icebergs, swamps, forests. The majority of protists that we're going to see, or the majority of protists that exist, are actually aquatic, but they can have a variety of habitats just because they have so many structures and so many different adaptations. So one of the first protists that we're going to focus on is the amoeba. The reason we're focusing on this one right now is because protists can be classified in a number of ways. And one of the ways you can classify protists is by looking at how they move, so their locomotion, you can say. Amoebas, they're a type of protist that will move using these extensions called pseudopodia or pseudopodium singular. So it's almost like they're crawling. They extend part of their membrane in the direction where they want to move and then basically pull themselves forward. So pseudopodia kind of means like a fake limb. It doesn't really have limbs. When it stretches out, it looks like a limb. It stretches it forward and then pulls itself in the direction that it wants to go. The pseudopodia can also be used to help the amoeba, for example, eat. So you can see here, these are the pseudopodia, and they're stretching to surround this food molecule here, or this could be a smaller organism, and it's basically going to surround it and then bring it into the membrane, bring it into that plasma membrane that surrounds the amoeba. So this is just one example of one way we can classify protists by the way of movement. So some organisms will move with pseudopodia, and one example of that is the amoeba that we see over here. So you should be able to spot an amoeba, you should be able to tell me how it moves, and you should be able to explain how it's able to eat using those pseudopodia as well. So amoebas use pseudopodia to move. Now this is not the only method of movement, there are some more. Another method of movement that some protists use is they use cilia. So if a protist uses cilia to move, we call it a ciliate. So for example, over here we can see the paramecium. It has many little cilia that it uses to move around. And they're just small hair-like extensions that surround the cell. So they're more numerous but also shorter than the flagellum. And there are some protists that move with flagella. And so the flagella, they are longer. You can see here we have a flagellum waving back and forth to help propel the cell forward. An example of a protist that moves with flagella is called the zooflagellate. And this is an example of a heterotrophic protist. So it can't make its own food, it relies on eating other things, but it has flagella in order to move around. So we saw that some protists are classified based on their movement. So for example, ciliates will move using cilia, and these are some great examples of ciliates over here. So if I were to ask you to explain how protists can be classified, you could say movement. You could give me the three main categories, so ciliates, for example, or some will move using flagella, or some will move using pseudopodia. You should know a good example of each one. So know an example of a ciliate, know an example of something that moves with flagella, and know an example of something that moves using pseudopodia. As soon as you spot it, you should be able to identify what it is. And so these are some of the examples that you should focus on and that you can take a look at in terms of their movements and how they propel each other. You should be able to look at D, for example, and know that it's a paramecium. You should be able to take a look at this organism here and give me some kind of reasoning in terms of how you think it would be classified. So you could see little cilia, for example, and you should know it's a ciliate, and this is the example of the Stelonchia that we saw in the previous slide. So again, please make sure you go over these different examples and read through them in your textbook and understand that this is the example we use to illustrate protists that move using different mechanisms. Another interesting category that you might see sometimes for protists is, we're still talking about being based on movement here, but some of the protists lack the ability to move. So they don't have flagella, they don't have cilia, they don't have pseudopodia, they just don't have the ability to move. So they rely on other things to move them. So they rely on what's called passive forms of transport. So for example, water currents might move them, wind might move them, some animals might move them. So the Plasmodium over here, it's a protist that causes malaria, and it doesn't really move itself, but it's carried around by mosquitoes, which are responsible for many deaths worldwide. So many of us are scared of these big, powerful animals like sharks, lions, and tigers, but in reality, these very dangerous ones are the mosquitoes because they carry this Plasmodium, which causes malaria. So here we can see just some questions to help you check your understanding as you go through this section of the PowerPoint, particularly in terms of the habitats that protists are found in. Can you define a protist? Can you draw diagrams of protists that we studied and how they move based on the classification that we discussed? And then here we have the big ideas that we saw in this section of the PowerPoint. So now we're going to move on and go see other methods of classification for protists. So protists can also be classified based on how they reproduce. So there are some protists that will reproduce using asexual methods, like binary fission, which we saw in bacteria. Essentially, let's take a look at a paramecium. That paramecium, if it does binary fission, it'll split into two paramecia that are identical to the original parent, and there's no diversity, but the thing is it's quick. And typically, the protist will do asexual reproduction when environments are good and when there's no need to have any variation and there's no risk of species going extinct. So very quick, just no diversity. Some organisms, some protists can do a bit of both. So for example, paramecia, they can actually generate some diversity in a process of sexual reproduction. Now, conjugation by itself is not really sexual reproduction. Conjugation is really just exchanging genetic material to create novel combinations, to create new combinations. So in reality, conjugation is actually something called recombination. So when we saw conjugation in bacteria, again, the conjugation process itself is not reproduction, but recombination. Now with paramecia, if conjugation is followed directly by some splitting, some binary fission, then we call that sexual reproduction. So binary fission alone without conjugation, that is just asexual, but some organisms can do sexual reproduction by combining binary fission and conjugation together. And that's a simplified way of looking at it. It's a bit incorrect, but it's a simplified way of looking at it that'll work for now. So students often take a look at this cycle diagram here. This is called a life cycle diagram. It's a reproductive life cycle for organisms. There'll be a separate PowerPoint that explains how to read these. I don't expect that you know how to memorize these, but more so that you know how to explain what's going on in the diagram if I were to give you the diagram. There might be some blanks in the diagram that you might have to fill out using other information in the diagram that's provided. So I care more that you know how to read these diagrams and understand the big idea as opposed to memorizing this diagram, because there's a lot here. So let's take a look at what happens. So here we have two paramecia. They're compatible mates. That means they are able to mate with each other. And when I say mate, they're not really going to reproduce, but they're going to switch some genetic material up. So they're going to join each other over here in this process of conjugation. You'll notice there's a little nucleus here called a micronucleus, which I'll speak to more about later on. That micronucleus is essentially a nucleus that's responsible for reproductive purposes. This is the macronucleus, the bigger nucleus, and it's responsible for every other day-to-day activity. But what I want you to notice is that meiosis will happen, and if you look at the picture carefully, after meiosis happens, each micronucleus of the paramecia, the two different paramecia, there's now 4 of them. So meiosis happened for the micronucleus, and I would expect you to be able to make conclusions such as this. If this is a diploid micronucleus, after meiosis, what would the ploidy of the micronuclei be? You would guess haploid, because meiosis halves a ploidy. So you should be able to come up with something like that. Don't memorize it, but figure out what the ploidy would be based on the keywords that are provided in the diagram. So now you have these 4 micronuclei in each of the paramecia, and some exchange is going to happen. So some of them break down, that's fine, but the ones that don't break down, they're going to exchange some genetic information. And now we have paramecia that go both ways. So now they've gone their separate ways. This one goes off and does something else, and this one over here, we're going to follow this one right now. It has 2 micronuclei from different sources, one from itself and one from the other one that left. We're going to have a fusion event that happens, and even if I don't define that, you should know what that means. Take a look at the pictures before and after. 2 micronuclei here, 1 here. Micronuclear fusion, the 2 micronuclei combine into 1 new micronucleus. So what we just described over here in the green, this process itself is not reproduction part, but actually conjugation part, which is just the exchange of genetic information. Now that we're going into the blue part here, this is our reproductive part. So we have our nuclei fusing together, and now we're going to have essentially some rounds of division, cytokinesis, or you could say binary fission for now, just making copies of this newly recombined cell, and then the whole thing can happen again. So the idea is that when you have conjugation followed directly by this reproductive, this making more of itself process, this is sexual reproduction in paramecia. If you just had this over here happening by itself, that would be the asexual reproduction process alone. And there's no variety, no diversity, usually during good conditions when there's no risk of things going extinct. Usually sexual recombination will happen under more harsh conditions. For example, when there is not enough food around. So again, don't need to memorize all of these life cycles. You need to know how to read them and understand the general idea behind it. The big idea in this diagram is that conjugation is happening, exchange of genetic information between two compatible paramecia. Then after the exchange happens, they go their separate ways, and then their reproduction happens in the blue part. You should know what a micronucleus is. You should have a good idea as to what's going to happen from picture to picture based on the keywords that are provided within. Don't focus on memorizing, because chances are I could give you a diagram that you haven't seen before, and you need to kind of figure out and explain what's going on with all the keywords, or most of the keywords provided to you. And that can happen during a test, a quiz, an interview. Here we can see a picture of just pure binary fission, pure asexual reproduction in a paramecium. So you have your paramecium over here, stuff is replicating, and it splits into 2 daughter cells that are identical to each other and to the parent cell. So usually under conditions that are not so harsh. So Here we just have the difference highlighted between macronucleus and micronucleus. Macro meaning big. It used to be called the meganucleus, but this is our macronucleus over here, and it's responsible for basically everything but the reproduction functions. So, for example, metabolism and other everyday tasks. But the micronucleus, the small one over here, that's the one that's involved in the reproduction and the conjugation processes of the paramecium. And just a few other important terms you should notice: cilia, making this a ciliate. You'll notice, and this is an interesting one, this is called the oral groove. It's kind of like the mouth of the paramecium, and food can enter here and enter the membrane. And here we have an anal pore. It's basically like the butt of the paramecium, and then waste can come out this way as well. And this is something very interesting. We'll talk about this later. It's called the contractile vacuole. So when too much water enters the protist, it runs a risk of bursting and diluting many of the ions in here. So what happens is this will absorb some of the water and then spit the water out later on, so that not too much water can enter and damage the cell. So some protists can do both sexual and asexual reproduction depending on the environmental conditions, so they can kind of switch it on and switch it off. Again, I don't expect you to memorize all these reproductive cycles. I just want you to get the big idea behind it, and if I give you this picture, could you explain what's going on? Could you explain maybe where the asexual one is, where the sexual one is, and use some evidence in the picture to support it? So you can see over here we have an example of an algal cell, which is an example of a protist, and this specific one is a chlorophyte. And we can see this side here is the sexual reproduction side. And sexual reproduction for these organisms usually happens under harsh conditions. So for example, when there's not enough nutrients around. So what you can see here is we have a mature haploid cell. And it's going to go through a process where it's going to meet another mature haploid cell, and fertilization will happen. So if you see this keyword here, I would expect you to know that if we have a haploid gamete, after fertilization, it becomes a diploid. So there's a zygote in there, and it's now a diploid. And that zygote, the interesting thing that's going to happen here is because there's harsh conditions, it's going to form this protective coat to protect it from the harsh conditions. And it's going to stay dormant for a little bit. So it's basically not going to be active for a little bit. After the dormant period, once those harsh conditions are kind of are reduced a little bit, what's going to happen is you're going to have meiosis produce some haploid individuals again, and they'll be released from that protective casing, and then they can go back, and if there are still some harsh conditions around, well, then they can just do some more sexual reproduction to make more protective layers. So the general idea is that under harsh conditions, this guy will combine with another one, make a diploid organism through the process of fertilization. The zygote will make a hard shell. Meiosis will produce the haploid individuals again, and then those haploid individuals will be reduced from the hard shell, and then if there's still harsh conditions, we stay in this cycle. So it's interesting to see that these individuals are actually haploid individuals. So we are used to seeing diploid individuals, but in protists and other organisms, you can have haploid individuals that actually live and survive and are the actual individual themselves. So main idea here is that some can do both sexual and asexual depending on the conditions. Under harsh conditions, this chlorophyte will use the sexual reproduction method. And again, understand what the keywords would mean if I gave you this picture. Under good conditions, where the conditions again aren't harsh, typically this chlorophyte is going to use the asexual reproduction method, and you can see this part of the cycle here. So basically it just goes and makes copies of itself, identical clones, in this cycle over and over again as long as conditions are fine, and they remain haploid because it's just an asexual reproduction method. Diploidy will not change. So it's important to focus on the color coding scheme, on the keywords that you see here, and understand maybe why this organism has a choice and when this organism can use asexual versus sexual. Another type of reproductive method is called the alternation of generations method, and this is typically found in multicellular protists. The other examples we saw earlier were in unicellular protists, the chlorophyte and the paramecium. So some examples of multicellular protists are brown algae. But you can also see this alternation of generation methods in plants as well, which are not protists. They're a different kingdom, but again, we can see this method in plants. So the main idea behind alternation of generations, the thing that really makes it unique, is that in this reproductive cycle, you have a multicellular haploid stage and a multicellular diploid stage. You have both haploid and diploid multicellular stages. So this is a simplification of the alternation of generation over here. Let's look at this diploid organism here. So this is a diploid organism, and it's multicellular. You can see it by the big circle. This diploid organism that's multicellular will go through meiosis, and it will produce some haploid organism. Now, through mitosis, this haploid organism will become multicellular. And so here we have a haploid multicellular organism. So notice we had our diploid multicellular organism, and then later in the cycle we have a haploid multicellular organism. Again, you should pay attention to the key words here. So for example, you can see that after fertilization, if I have N, you would expect 2N here. So if I had this blanked out, would you be able to tell me that after fertilization I have 2N? Would you be able to decipher what's happening in this process? But the key giveaway at this is alternation of generation is that you see multicellular haploid, multicellular diploid. We'll take a look at a more specific example on the next few slides, but that's just the general idea behind it to identify it. Some key words to really focus on when you're studying this are the words sporophyte, gametophyte, and spore, and you can see that on page 65 of the Nelson textbook. Humans don't follow alternation of generation. Our haploid stage is unicellular. It's a gamete, a haploid gamete, and you don't see a multicellular sperm walking around. It's just a unicellular haploid gamete. So when you see these multicellular haploids and diploids within one life cycle, you know you have alternation of generation. You're basically alternating between haploid and diploid multicellular phases. So here we can see an example of alternation of generations in the brown algae. And so here we have the terms that I told you to focus on for alternation of generation. Let's look at the sporophyte first. So the sporophyte is a diploid organism that produces haploid spores. Okay, so here we have a sporophyte. This is our mature sporophyte. It is a diploid organism. It's multicellular, and what it's going to do is, and notice it's diploid right now, it's going to produce these things called spores, and those spores are haploid. And now these spores, they start off as single cells, but eventually those spores will grow into these multicellular haploid structures, and those are called the gametophytes. So the haploid cell that was produced, that spore, becomes a gametophyte, and the gametophyte now becomes the haploid multicellular part of the alternations of generations. Now this gametophyte is going to produce some gametes. This gametophyte here, let's call it a female one, is going to produce other gametes, the egg cell. The male one, the male gametes, will go fertilize the female gametes, and that will bring us back to our diploid stage. We have a zygote that's diploid, and over time, through mitosis, it becomes a mature sporophyte again, which is the diploid, the multicellular diploid stage in the alternations of generations. Now I want you to get things like this. If we have a zygote over here that's diploid, and you see it become multicellular, you should know that was through mitosis. So whenever you have a single cell become multicellular, that's typically through the process of meiosis. If we see going from diploid to haploid with this color coding here, you should be able to figure out that that's probably meiosis. If you see, again, same thing here, we have a haploid unicellular become multicellular, that's probably through mitosis because we didn't change the ploidy and it became multicellular. So usually when you see something become multicellular, that's through mitosis. The main point here is that we have a diploid multicellular stage and a haploid multicellular stage. We have some sexual reproduction happening here as well, and this is called the alternation of generation. We see it in plants, but also in protists like brown algae. So these are the representative groups of the protists that I expect you to focus on. You should know what their main energy sources are. You should also know some key features about them. So if I ask you for an example of a protist, you should know, you should be able to say maybe a diatom, and that it's a photosynthetic organism, and that it's unicellular, and that it can move around by gliding, and that it's, you should have some general idea of what these organisms are, and you should also be able to identify them in pictures because they're on your slides and also in your readings. So you can see here are the various examples of cells and protists that you should be able to identify, and then I'll show you some multicellular ones that you should be able to identify as well. So not only do we see that protists could be classified based on their method of reproduction, but they could also be classified based on how they move. Another method of classification is the lifestyle of the protist, or in other words, how similar they are to other kingdoms. So you'll have some protists that are animal-like protists, they're similar to animals, some protists are plant-like protists, they're similar to plants, and some protists are fungus-like or fungal-like protists, they're similar to fungi. So you can see over here the amoeba, which is an animal-like protist, the paramecium, which is an animal-like protist. You can see the euglena, which is a plant-like protist, and I bet you can already point out the feature that makes it plant-like. It is the chloroplast. That's right, very good. And then we have over here a slime mold, which is like a fungus. Usually when you hear the word mold, you think fungus because most molds that you hear of are fungi. But when you see the slime mold, it is a protist that is like a fungus. And so we're going to go and take a look at what makes these protists here animal-like, what makes this plant-like, but we've already kind of seen it, and what makes this fungal-like. In terms of animal-like protists, the special name for them is protozoa. And again, think protist from the zoo, like animals that you see at the zoo. The main thing that makes a protist animal-like is that it's a heterotroph that has to go capture and ingest its food. So it's non-photosynthetic, it can't make its own food, it does not have chloroplast. So like us, we are heterotrophs, humans, we have to go capture our food and then put it in our mouth, and that's ingesting. So here we can see the amoeba. It uses the pseudopodia to reach out, surround the food, and then bring it into the cell membrane, and that's ingesting the food. Here we can see the paramecium, another animal-like protist. We can see the oral groove. Food goes into the oral groove, and once it goes in, it's now surrounded by this little membrane sac called the food vacuole. We break it apart, use the parts that we need, and then we can release it through the anal pore over here. But the main idea is that these animal-like protists are animal-like because they're heterotrophs. They have to go capture and ingest their food, and the other name for them is protozoa. So paramecia and amoeba are classified as protozoa if you're looking at the lifestyle way of classifying protist. So again, make sure you do know how to identify the different organisms, and make sure you know how to classify them as well. So the amoeba, you now know several ways to classify it. You can classify it based on its movement. You could also classify it based on the fact that it is animal-like protist. You should know other details about the amoeba. So on this slide here, I would know these details, what genus is part of, for example, that it's unicellular, that they don't have a definite shape, and that they move using pseudopodia, and that they're animal-like as well. Same thing about the paramecium. You should know that it is unicellular, it's a ciliate. You should know it's called a protozoa. Protozoa is basically the other name for animal-like protist. Think protist from the zoo. You see animals at the zoo, so protozoa. And you should know general areas where you can find these paramecia, so usually in the freshwater and marine environments, and know some of the different parts that we discussed, like the macronucleus, the micronucleus, its oral groove, its anal pore, contractile vacuole. So the basic general parts you should be able to identify and know what they are. So as I mentioned before, know how to label protozoa, again, which are essentially animal-like protists. Here I just have a formal definition of the contractile vacuole, but we'll take a look at more details of this in the next presentation. So we also have plant-like protists, and the main name we give the plant-like protists are algae. So the thing that makes plant-like protists plant-like is that they're autotrophs. They're able to make their own carbon-containing compounds. It can make their own food. Typically they're photosynthetic and they contain chloroplast, kind of like plants do. Many of them might have cell walls, similar to plants, but some they don't necessarily have to. The main thing is the fact that they're autotrophs, but some could have cell walls. Just because they have a cell wall doesn't necessarily mean they're plant-like. They could be maybe fungal-like, but again, autotrophs is that key defining feature. Many algae are actually unicellular. Some will live in colonies, and others can be multicellular. In terms of the reproduction, most algae can do both asexual and sexual mechanisms. We saw the example of the chlorophyte earlier. We also saw some algae that could do alternations of generations. And usually these plant-like protists, if you want to further classify them, you can classify them based on their cell wall composition, maybe the types of carbohydrates they have using their DNA sequences, and also what type of photosynthetic molecules they have. So there's certain pigments that allow them to do photosynthesis in slightly different ways. You can classify them based on that as well. So the euglena, this is an example of a plant-like protist. So this one doesn't have a cell wall. So like I said, you don't have to have a cell wall to be considered a plant-like protist, but it does have chloroplast to allow it to do photosynthesis. So when you take a look at this organism here, you'll see the chloroplast is that kind of main defining feature, and you can see it in more of a real life or a real live image over here as well. Now this one's a bit difficult to classify because it actually is an autotroph. When there's light around, it will do photosynthesis, but in the dark it could actually become a heterotroph and actually start absorbing nutrients. So we could actually say this one here is a mixotroph. So you were asking for some examples of mixotrophs, so now we see a great example of a mixotroph over here. Another very interesting feature of these plant-like protists, or example of plant-like protists, are phytoplankton. So when you hear the word phytoplankton, those are protists. So plankton are usually microscopic organisms that exist in aquatic communities. And phytoplankton, they're often the producers that form the base of the food chain in those aquatic food webs that you studied in grade 9 science. And so if you get rid of those phytoplankton, then your food web can be damaged or affected in serious ways. So this is another example of a plant-like protist that you've actually heard of before, but you didn't quite think or know that it was a protist. Some other examples that you should know are diatoms. So diatoms are pretty unique in how they look. I mean, as soon as you see this, you'll know it's a diatom forever if you continue in biology. So most diatoms, they're a major group of algae, and they're the most common phytoplankton that you see. So phytoplankton, there's not just one type, there's many types, and diatoms are the most common ones. Most of them are unicellular, but like I said, some can exist in colonies, and they're producers, so they make sugars for the food webs using photosynthesis, for example. Other examples of plant-like protists are dinoflagellates. Dinoflagellates are quite interesting. So they do have a cell wall made up of cellulose, and the reason they're called dinoflagellates is because, well, they have two flagella. So they have two flagella. You can see the flagella over here. And the majority of these dinoflagellates are photosynthetic organisms. And here you can see some examples of dinoflagellates. Whenever you see this red in the water, I want you to read up on that. Harmful algal blooms. We call them HABs, and we don't mean the hockey team that's rival to our Leafs. They create this. These are protists that create this red color in the water because they have these carotenoid pigments that give them that color. There's some other dinoflagellates that are bioluminescent. They basically glow and create their own light. And so there was this company, and I think it's still around, you can actually order one, that created these dinopets where they send you these dinoflagellates, and basically when you shake them, they'll create light and bioluminesce. And you can actually see that in nature as well. Here we can see an example of some dinoflagellates actually being shaken up by the waves, causing a chemical reaction. You don't have to memorize a chemical reaction, but that chemical reaction will eventually lead to glow and flashes of light in the water. So that's caused by protists, by dinoflagellates, which is quite amazing to see. Other plant-like protists that maybe you didn't realize, for example, if you're a sushi fan, that seaweed that you see, that's an example of protists. So red algae, even though the seaweed is green, it's actually classified as a red algae, and that's nori specifically. Many of the seaweed products are actually used in food additives and toothpaste, so we can see some great examples of some positive and good protists here, unlike the negative protists we saw that can cause diseases in the beginning of this presentation. Red algae, another important thing about them is that they produce this thing called agar, and you can create these little dishes where you can grow bacteria to study them. The bacteria will grow on the nutrient agar medium in that little home called agar. So some great applications of protist over here. Again, we saw red, green, and brown algae, and these are examples of multicellular protists. So a lot of the other ones we saw were unicellular, these are multicellular, and these are seaweeds. So one important area of confusion that I'd like to bring to your attention is the classification of green algae. So they were once all considered protists. And many sources still do consider them protists, but now because of fossil evidence and DNA evidence, some of the green algae are starting to become considered part of the kingdom of plants instead. So some green algae are actually considered sort of the oldest group of plants that exist. So if you take a look at this phylogenetic tree here, here we have a plant ancestor, but the outgroup to these plants is this green algae over here. Typically, some of the multicellular green algae are slowly starting to phylogenetically become considered part of the kingdom of plants. It's not really written in stone yet. It's really a hot topic of debate, but there's really quite a few similarities between some of these green algae and plants. In fact, some of the green algae are more similar to plants than they are to other types of green algae. And the evidence that we use is the cell walls. They're very similar to some of the plants. Also, the photosynthetic pigments, which are the molecules that many of the plants use for photosynthesis, are very similar in some of the green algae when you compare them to plants. So some of the green algae are reclassified by some biologists as plants. Your textbook does not quite go over that, but sometimes you will read up on that debate of green algae being a plant or not being a plant. For the most part, we're going to consider green algae protists still, but I do want you to know that it is one of those debates that will continue going on until more evidence is found to lead to a proper conclusion. So you should know how to label algae cells. So for example, you should know how to label this cell over here, know why it's a plant-like protist, specifically focusing on the idea that it has a chloroplast, and you should know what the other parts are as well. You should know how to label a euglena, another example of a plant-like protist, again focusing on the idea that it has a chloroplast over here. Might be a good idea for you to read up on what an eye spot is, because you've maybe never seen that part before, and hopefully you're getting an appreciation for, yes, all these things are cells, but there's so much diversity in these cells in terms of their shapes, in terms of their adaptations, in terms of these things called contractile vacuoles. Why do all these different parts exist? What advantages does it give the cells in the environment? So hopefully those are all questions you're just asking yourself as you're going throughout this unit. And finally, we get to the fungal-like protists. The main thing that makes fungal-like protists similar to fungi is that they're heterotrophs, but they feed mostly on decaying matter, kind of like fungi do. So fungi will decompose things, so will fungal-like protists. So they're heterotrophs, and they will feed on decaying matter. Unlike a real fungus, the fungal-like protists, they'll actually contain centrioles, which a real fungus does not contain. And also the fungal-like protists, they don't have chitin in the cell walls. True fungi will have chitin in their cell walls. In terms of reproduction, they can reproduce sexually or asexually, depending on situations, and some can be unicellular and some can be multicellular. Some great examples are cellular slime molds and water molds. Usually when you hear the word mold, that refers to a fungus, but when you see cellular slime mold, this is a fungus, a fungal-like protist. and this one's a decomposer specifically, and it will mainly decay matter that's on land, so it's a land type decomposer. And water molds, again, not a fungus, because you see the word mold there, it's a fungal like protist, and it will decompose things, and typically it will decompose dead plants and other matter in water as opposed to on land, like the cellular slime mold. So here you can see examples of cellular slime mold, and here you can see example of a water mold that's decomposing a dead fish. This organism over here is a species of fungal-like protist that actually caused a great potato famine that killed nearly 1 million people, and that happened in 1846 to 1847. It is a water mold, and it basically wiped out or decimated potato crops in the country, which was an essential part of their diet back then. And it also affects tomato plants too. So there can be some negative protists, again, as I mentioned. But one interesting, let's say, cure to this or remediation to this is genetic engineering. So some GMOs have been made that can resist this water mold. So they took a gene from a related potato plant and they put it into other potatoes so they could become resistant to this water mold. So again, when people say that GMOs are bad, well, are they really that bad? You need to kind of question that, because there are some benefits to these GMOs. And here's an article about that genetic engineering process. So here we have the summary of the concepts that we've seen. Remember, protists they vary quite a bit. They're organisms that don't fit into the other kingdoms. They're eukaryotic. You can classify protists based on how they move. So we saw pseudopodia, cilia, flagella, or no movement. You can classify them based on how they reproduce, sexually or asexually. Some can do both. You can classify organisms based on their lifestyles, protists based on their lifestyles, the plant-like ones, which are autotrophs and have chloroplasts to do photosynthesis, animal-like ones, fungal-like protists. So you should know the general classification methods for protists. The common examples that we saw in this presentation are the ones you should focus on and know how to label. And these are just some concept check questions that you should do in addition to the textbook questions that have been provided to you or that are posted online. Here are some more concept check questions, and it's just another summary of the big ideas worded differently in case there's some things that you missed out on. Understand some of the benefits of protists, and also some of the harmful impacts that protists can have on organisms and on us. This slide here is a great example of a career that you can have if you're really interested. If you're very interested in protists, you can be a protistologist and you can study protists for a living. This is a great research assignment that you can do to have some examples of the different protists that we saw. So you can research some video clips for ciliates, pseudopodia, and flagella type protists. You can look up some of the life cycles that we discussed. So you can look up, for example, Vivax plasmodium and see its life cycle and how it's a parasite, which means causes harm. And then you can take a look at the potato blight example that we saw earlier and find out which protist is responsible for it, but we already answered that question. And you can also take a look at the GMO I discussed earlier and find out which plant scientists used to make the GMO for the potato plant. So a little bit of a research assignment that you can do if you're a protistologist and you study protistology. You will be doing research like this over here. So this is our summary table. Our summary table keeps getting larger and larger and larger. So far we had seen the bacteria and archaea in our table, and now we added protists to it. So you should know all those general features and the main organisms within the table. Read through your textbook. That'll be the main focus, along with the PowerPoints that are here. When I put videos online and articles, you can quickly watch those videos to enhance your knowledge. I won't expect you to memorize everything from those videos unless I specifically say so. Some of the articles, it's good to maybe know the big idea of it. So if I ask you for a specific example you can give one, but don't go and memorize everything in the articles. Focus on the PowerPoint, focus on the readings in your textbook, but you got to focus on something, so make sure you're doing the work in the readings. It's the only way you'll really understand this material. So here are some homework. Here's some homework you can do. The handouts, which are posted online actually, and the review questions for the PowerPoint and the readings. These are the readings for the protest sections, and there's some videos you can watch in case the readings and the PowerPoint does not suffice and does not really help you understand what you need. And then we have some related other related videos for protest over here as well.